Hi, welcome to the Bioinformatics Chat. Today I'm speaking with Romain Lopez, a PhD student at Berkeley, and Gabriel Misrachi, a master's student at École Polytechnique. And we will be talking about scVI, a model for single-cell sequencing data. Guys, welcome to the podcast.
Hi, Romain. Thank you very much for having us in the podcast. It's very exciting.
Hi, Romain. Very nice to be in the podcast. Very excited to talk with you, and hi to our listeners.
So I think we've covered single-cell sequencing in general a few times on this podcast. And I think by now people are more or less familiar with the concept. But I'm curious, when you started to work in scVI, and it's been a few years already, what was the landscape back then? I think there were quite a few tools already to analyze single-cell sequencing data. What could one do at the time, and what couldn't one do? And what was the motivation for starting to work in scVI?
That's a very good question. So basically, I started thinking about scVI when I was visiting the Harvard Medical School, especially the Systems Biology department with Alon Klein. And over there, I was basically looking at how people were analyzing these data sets. And I saw that the main streamline was basically taking these matrices of counts, normalizing them with different ways, and just trying to find the best that can work, using a PCA, and then trying to visualize and cluster the data. And I could see that there were other algorithms that were very tailored to the area. So let's say 3-4 years ago, single-cell transcriptomics was already very crowded in terms of computational methods. But most of the methods that were either very simple or tailored to the data, but not scalable. And also one other thing is that there were a lot of different tasks that you would like to perform with single-cell data. For example, so you would like to normalize, you would like to, a common theme is also to impute the zero in the matrix, or you would like to cluster, find cell types, perform differential expression. And for all these type of tasks, basically you would have a different algorithm and even a different software. And so it was kind of challenging to understand what were the different assumptions of all these algorithms. And so the idea of scVI comes into this dual aspect. First of all, we wanted a method that was tailored for single-cell data, but that was very fast. And second, we wanted to achieve what I like to call model consistency, which is this idea that with the same graphical model, we would like to perform maybe not extensively all the tasks, but at least a very large subset of them. And so, for example, for the scalability, we were really interested in this 1.3 million cells data set from 10x Genomics. And we were also seeing that if we were able to build an algorithm that could correct for batch effect on a very, very large scale, would enable very large studies and working with the human cell atlas data or some other kind of consortium. And second, we worked on how to design this graphical model so that you could perform all these tasks with the same software, but also with the same model somehow. And so on the scalability, one of the main results of our paper, and so a lot of papers after, in the same time than us, that were also developing these VAEs, came up with the same scalability, which is essentially that you can analyze this 1 million cell data set in around an hour. And second, in terms of model consistency, we arrived to a regime where we were able to perform a certain range of tasks, and we were able to outperform the state-of-the-art models in this task.
one thing I wanted to clarify, you mentioned the graphical model a couple of times.
Yeah.
And lest people think that this has anything to do with graphics. It doesn't, right? It's a specific statistical kind of model.
Yeah. So it's an idea borrowed from the meeting of like statistics and computer science, it's a family of probability distributions. So it's essentially a modeling tool for Bayesian models. Yeah, and you have this graph. Why? Because in the graph, all the nodes are random variables. And all the edges are encoding conditional independence. So it's a modeling tool, essentially.
And so you were dissatisfied with the variety of tools, each tackling a different aspect of single-cell analysis. And someone else could have written a single tool which would do like all of these different analysis, like have many modes of operation, and like using similar statistical assumptions for them. But you went even farther than that, and you developed sort of a super model to rule them all, so to say. And can you talk about like what scVI is and what it does?
Yeah, very good question, Romain. What scVI does and how one might use it. For starters, scVI is designed for inference on single-cell data. One of the tasks concerned is dimensionality reduction. So often single-cell data will have, for a single cell, thousands of genes expressed. So you'll have numbers for each cell. You have 1,000 of numbers for each cell. And these numbers can be hard to use for interpretation and analysis. So what you want is to get a lower set of numbers, a smaller set of numbers to capture the same information. So what we do is build what we call a latent space that is basically containing these say 10 numbers for each cell instead of 1,000 gene counts. This representation of single cell data is all the more useful because it is designed within the scVI graphical model to only capture the biological signal and not the technical noise. So one of the other tasks that one can perform is gene count imputation. So one can impute either the observed counts or a normalized, shall we say, version of the counts. The normalized version is especially useful because, again, it stems from the latent representation that we were talking before and only contains the biological signal, which is disentangled from the technical noise or the technical zeros one usually observes in single cell data. These normalized counts are especially useful for downstream tasks. One of these tasks, for example, is differential expression. For instance, this means that we want to find for two specific population of cells marker genes for one against the other. So we're going to use the normalized counts that contain the biological signal to try and figure that out using Bayesian hypothesis testing, mostly. Another task that scVI can perform is dataset harmonization. So this basically amounts to removing the batch effect from a set of data sets that you have. And if those data sets are initially close enough, the objective is for the actual cell types to overlap in, for instance, the latent space that scVI builds. Also, so we kind of covered what scVI does in a very brief and non-exhaustive way. And now let's talk about how it does it and how one can use scVI. So very importantly, we put a lot of work in our code base that is available open source on GitHub, and it's tens of people all around the project that have worked on it and have made a great effort to make it as useful as it is right now. And we make it a point to keep it up to date and put all our models, even the most recent research, on there. So please take a look. Feel free. You'll find some interesting stuff. Importantly, we have a lot of ready-to-train datasets available in the codebase. So if you want to try it at scVI, we have all of your 10x and etc. Available, ready to go. Just a one-liner of code to get them. We provide lots of notebooks, an example ranging from our basic tutorial to a full tutorial on our hyperparameter tuning feature, and also on our latest model, scVI, for missing gene imputation. And finally, we provide samples of code to interface scVI with ScanPy and the data format, as well as the LoomPy data format and libraries, which are well-known single-cell libraries that are very useful. Additionally, the code base is built so that one can, as easily as possible, develop new tools on top of scVI. This means that we have an API oriented towards machine learning researchers, which basically amounts to having a consistent terminology with machine learning frameworks, which makes the code base easy to take on and easy to read and understand. And we provide general abstractions allowing for fast prototyping of new model ideas or trainer research ideas in general.
So indeed, yeah, scVI can do a lot of different tasks, but it doesn't come exactly for free. For example, the price of having a pretty complex model is that it needs a lot of data to fit, and there are a lot of instances, especially in the early setting of single-cell RNA sequencing protocols, for which not a lot of measurements, which means cells, are available. And so for this, using a lower complexity model, such as PCA or alternative method, will work better than scVI. So that's kind of the price to pay for having such a model.
Right. So if I understand correctly, the core idea of scVI is to use this statistical model that we will talk about a little bit later to basically reduce the very high dimensional space of the gene counts and gene expressions, which has thousands or tens of thousands of numbers per each cell, to this very low dimensional but information rich representation. And then you can do all these different things. They become much easier. Things like differential expression and imputation and harmonization, they become much easier once you reduce the data to this small representation. And if you use scVI, do you ever sort of interact with that low dimensional representation? Can you ask for it?
Yeah, so you're right. It's an important part of scVI to provide this Lin code of the data, which is rich in information, and that is disentangled from some part of the technical noise. And indeed, it is possible to access it using the code that we provide. You just call the method of our posterior class, which represents the posterior distribution that you obtain after training the scVI model that allows you to perform inference, and you call this and get your the latent representation for your data.
Right. So you can do a lot of things out of the box. You can do a lot of sort of standard analyses, but if you need something custom.
Yeah, you can completely access every stage of the graphical model, every node. So you can sample the latent space, the normalized counts, the imputed observed counts. You can sample all of these nodes that are in the graphical model representing scVI.
So when one uses scVI, so far we're talking just from the user's perspective. We'll move to the developers or statisticians' perspective a bit later. But from the user's perspective, you run this tool on your data. So what kind of data does it accept? Does it accept like a CSV file of expression counts, or?
So we have interfaces for pretty much any format that you can think of, be it CSV, H5, LumPy, really any kind of data. And we provide ready-to-train API for some of these data sets. For example, the data sets that are hosted on the 10x website can be imported directly using only a single line of code that will download and pre-process the data for you. So really any kind of data. If you have some type of data that can't be used, please mention it on the GitHub repo. We'll make it available as soon as possible.
That's pretty cool. And as with almost any statistical model, there are certain parameters that you have to set. They're called hyperparameters. They're not estimated from the data. They are set by the user, by the statistician, that basically determine the specifics of the model, the specific form of the model. Gabriel, I know that you recently worked on automating that away. So even though you, like in theory, you need to set them based on your expert knowledge, in practice, very few people have that expert knowledge. And so you developed a method to basically find the best hyperparameters. Can you talk a little bit about that work?
Of course, yeah, with pleasure. So yeah, this is one of our most recent tool, but let me maybe give a bit of a bit more context to our listeners. So yeah, what are hyperparameters? So indeed, as you said, with a machine learning model, you have two different types of parameters. You have the learnable parameters that are, for instance, the weights of your neural network. That will be optimized using, for example, your stochastic gradient descent method or any training method, and so these are parameters that you can differentiate your model against and optimize this way. But you also have a set that we call hyperparameters. That are essentially the parameters that are not differentiable against and that are set by the user, as you said. These parameters include, for example, the parameters of your optimizer, such as the learning rate, or more importantly, the model architecture. So for a neural network, it could be the number of hidden layers or the number of hidden neurons, etc. So these are very important variables. So the motivation to perform an automated and optimized setting of these hyperparameters is as follows. The first point is to obviously maximize performance of the model. So for starters, you choose a metric to optimize, and then you perform this hyperparameter optimization process to get the best set of hyperparameters with respect to that metric. In our case, we tend to do that to maximize data fit, which is in our case negative marginal log likelihood. And we believe that this should help for most of downstream tasks. The other motivation for this new tool is to have a way to perform principled model comparison with scVI. Because such as Hugh and Green mentioned in their paper on the importance of hyperparameter search for VAEs, researchers tend to spend more time fine-tuning their model than those they compare themselves to. So, and in general, this is then detrimental to the comparing process, especially if competing methods have a high sensitivity to hyperparameters. And this is often the case. I mean, for example, on image classification task, the same CNN can be trained to near perfection as well as complete randomness, just by changing the hyperparameters. So this is the main motivation for the development of this new tool.
So you want to avoid a case when someone develops a competing method and they claim that they outperform scVI, whereas in practice they spend a lot of time tuning their hyperparameters, whereas like yours are set to some arbitrary values, and when the setter of the hyperparameter is supposed to be an expert, that particular person is not an expert in scVI. The only thing they want is to compare it to scVI. So they just set some random hyperparameters. It doesn't perform well, and that's good for them.
Yeah. I mean, yeah, I'm not saying that's done on purpose. It's just a natural behavior to spend more time on your own model than the one you're trying to compare yourself to. So,
right.
Yeah, in order to alleviate this problem, we just provide an automated framework to perform hyperparameter setting with scVI. Yeah, as you mentioned, it's exactly that.
And of course, the end user also benefits because she doesn't have to bother and think about these hyperparameters and have her research jeopardized because she may not have picked the best values.
Yeah, of course. And yeah, the user can maximize their performance with respect to the metric of their choice, and that's good for them because they don't have to spend any time trying out combination of parameters, and which can be a very cumbersome and long process.
Yeah, so what does this look like from the user perspective? Do they just type like --auto and it just does the magic?
So basically, we have a default way of performing a hyperparameter search for scVI, so using that default behavior amounts to 5 lines of code. So you pick a data set, you pick a model, pick a trainer. Trainer, I mean that we have some models, some scVI models are completely unsupervised, some are semi-supervised. So you pick one of those trainers, supervised or unsupervised or semi-supervised, and then you just call the tune my model method and give those as input. Maybe give a budget on how many evaluation will you allow, and the process itself will output you the best set of hyperparameters for your query, knowing that in that case we provide a default search space for their hyperparameters. That means that, for example, we will allow the number of hidden layers in the scVI neural nets to vary from 1 to 5, and we have a set of defaults like that for every important hyperparameter in scVI. So that's the default behavior, and and you can customize this behavior in many ways. We detail this in our tutorial on the GitHub repo, but in essence, you can use a custom search space for your parameters if you want the learning rate to range differently than the default. You can just input that, and you can use you can even use custom objective function, which means that you can pretty much do anything. But more importantly, what I want to point out is that this optimization process automatically leverages every GPU available on your machine and is going to run in parallel. So this is a great way to speed up your hyperparameter search in a seamless and a completely automated way.
Sweet. And you also wrote a blog post about that, which we'll link to on the website.
Yes.
But let's dive into the technical details. So that magical statistical model that we mentioned a few times already, what are the sort of essential features of that model? How is it different from the models that have been proposed before?
So about the model, there are maybe a couple different steps we can take to explain scVI. I think explaining scVI from scratch in 2 minutes is quite a challenge that I can try to accept. But basically, so we can cut this down into small chunks. So scVI relies on the variational inference, and I will try to explain the basic idea, statistical idea of variational inference. And then also scVI belongs to a family of, it's a variational autoencoder. So I can try to explain a little bit what is an autoencoder and what is the rationale behind it. And then once I explain this, I will try to explain why scVI is not a vanilla autoencoder or not a vanilla variational autoencoder. So those are a lot of terms, but I hope that it breaks it down into small pieces. So first, let's try to understand what is an autoencoder. So an autoencoder is an old idea from neural network research. It has been used in many areas, and the main idea is let's say I have some I work with data. I'm in the unsupervised learning framework, which means I don't have any labels, I don't have anything to predict, and so might as well try to predict myself. So that's the basic idea. So for this simple scheme, you need two functions that are called neural networks, because in this case these are neural networks, so you take your input variable and you map it to an abstract variable, and then from this abstract variable you try to reproduce your input, and then you minimize a loss, which is the reconstructed input versus the original input. And here the abstract variable is called it's equivalent to a latent variable, or what we call the embeddings, all these type of objects. And it's supposed to summarize the information from the input into, preferably, most of the time it works better if the intermediary variable is low dimensional, which means it has a low number. And so this is an autoencoder, and it's linked to this literature of compression and all these kind of things. And for a basic example is that if the loss I was referring to is a quadratic loss, and the neural network are actually linear, so with 0 hidden layers, this is basically a PCA. So an autoencoder is just a nonlinear version of a PCA.
Right, and it sort of makes sense why this is a good fit for scVI, because this mirrors what we already described about dimensionality reduction in latent spaces. That's what you're trying to do. You're trying to reduce this gene space of the dimension 20,000 or something to a small dimensional space. And one way to achieve it is to have an autoencoder have this pair of neural networks that work together to reduce, to map the high dimensional space to the low dimensional space, and then the low dimensional space back to the high dimensional space to minimize the discrepancy.
Exactly. But there are quite a bit of challenges with applying these autoencoders to single cell data. And basically, because these autoencoders are a bit too simple, either you need to normalize your data beforehand, because otherwise the autoencoder will try to also predict the, let's call them confounded variables, such as the library size, such as the batch effects and all these things. And so if you just try to reproduce your input, but your input has batch effects, your latent variables are not going to take this. I mean, it's called the bottleneck also, the variable in the middle. are not going to be very interesting because they're going to contain the library size and they're going to contain the batch effects. And so somehow the reason, so scVI, for example, has like it's kind of, there are like two autoencoders, if you will, for the different variables. And the reason we need several of them is that we want to do this statistics gymnastic of separating out some different type of variation in the data. And so also, and because we didn't want to normalize the data beforehand, but we thought that the models should learn how to normalize it itself. That's why we stand out from these autoencoders, and we wanted to add a little bit more statistics to it. And so that's why we introduced this a Bayesian model and this inference framework. So now let me try to explain give a little bit of a flavor of what variational inference is. And so basically, in scVI, we designed this, so I was talking earlier about this graphical model in which we can enforce some variables to be conditionally independent or marginally independent. And so, for example, the way we design scVI, we would like this bottleneck I was talking about earlier, or this like low dimensional representation. We would like it to be marginally, so like independent from the batch, for example. So we would like it to be independent from whether it was sequenced from 10x or with SmartSeq2 or other technology. We would like it to be also independent from the library size. So we need to model the library size. And then we thought a lot about what kind of conditional distribution would fit to model the counts. And I can talk about that a bit later. But basically, once you have this model, you need to perform inference. But because the model we built is not as simple, and it doesn't satisfy some basic statistical assumptions that you need for what we call tractable inference, which means we can learn the model easily. Then we need to resort to some kind of approximate inference. And
now you're talking specifically about Bayesian inference, right? Because from the point of view of autoencoders, which are usually optimized like using a generic optimization routines, it doesn't really matter what your model is. You just optimize, you just fit it to the optimizer. But once you shift your perspective into the Bayesian inference, then there are certain models that are well suited for more like analytical inference. And you're saying once we add this all complexity and consider it from the Bayesian perspective, which means that we want to learn not just the point estimates of the parameters, but the posterior distributions of the parameters. Now it becomes expensive to apply something like Markov chain Monte Carlo to it.
Exactly. And so basically, where the variational inference jumps in is to exactly turn the Bayesian inference exercise into an optimization problem. And that's where it gets interesting, in which basically, so the way we would do Bayesian inference is to maximize the likelihood of the data. And this is something we cannot do. We would need to resort to Monte Carlo, which is very expensive. And so instead, what we do is that instead of optimizing the likelihood itself of all the data points, we can maximize a lower bound on the likelihood, which is something that is always less than the likelihood of the data. And there is a simple way to parameterize these things with an approximate distribution, which is the approximation of the posterior. And basically, this lower bound is going to be tight, which is something good. It's going to be very close to the data likelihood, if and only if the approximate posterior is equal to the true posterior. And the relationship between this basic statistical idea and the autoencoders is that so this has been described by two papers that describe VAEs from 2014. And basically, what you would like to do is, instead of learning a variational distribution for each of the data points, you're going to parameterize the parameter of the variational distribution by neural networks. And so all these networks that encode the variational distribution are called encoders. So as in the autoencoder, you had an encoder and a decoder. So all the variational inference, if you want, parameterizers, or the parameter for the variational distributions, or the encoders, and all the parameters for the generative model are in this decoder. And that's where we stand from autoencoder to variational autoencoder. And it turns out that even if in the original formulation of the VAEs, people would use only basically two latent variables, two variables, one for the observed things and one for the hidden things, and they would make inference over this type of object. In a scVI, we try to leverage this basic idea from Bayesian model to perform inference over this ideal model that would disentangle library size, batch effects, and that will retain only what we hope to be the biological information.
And speaking of Bayesian inference, I'm curious how you came to the decision to treat this as a full Bayesian model. So even though your model specification is in the form of Bayesian network, and you specify the generative model, still you didn't decide just to find the most probable point estimates of all the parameters, but you decided to perform this full variational inference. And I guess part of the reason is the connection that you mentioned with variational autoencoders. But what were are you a Bayesian by default, or were there specific considerations that moved you to use Bayesian inference as opposed to like point estimates?
Yes, that's a really interesting question. So basically, if you look at the tasks that we're interested in performing for a scVI, most of them would not require this idea from variational inference. And there is a paper that came out exactly in this. So a lot of papers that perform autoencoders or variational autoencoders came out at the same time on bioRxiv, maybe between 5 and 10 preprints. And one example from Fabian Teillet's lab, they built an algorithm called DCA, which is an autoencoder with the same type of loss that we use in scVI. But here you do not have this variational inference. And so with the algorithm, you can perform a wide range of tasks, but there is something that you cannot do. And this is actually why we use this variational inference framework. And what you cannot do is basically estimate the uncertainty of the parameters, of course. And and if you cannot do uncertainty of the parameters, then you cannot do differential expression. And so when we try to add more and more tasks to a scVI, we came to the conclusion that if we didn't add this latent variable to correct for the library size, and we didn't use this, we didn't have this posterior uncertainty, then we couldn't do differential expression. So that was one of the main motivations actually to use this these variational distributions and all this type of thing.
Okay, and while we're on this topic of differential expression, do you want to briefly explain how exactly you approach that?
Yes, of course. So basically, differential expression is defined as I have two sets of cells, and I want to find which genes are significantly different between these two sets of cells. And so there are different ways to think about it, but because we try to remove this library size and we try to remove this batch effect in a scVI, we are able in this graphical model to query, which means basically sampling. We're able to sample many values of, let's say, the mean of the negative binomial for each cell. And so, for example, when we have a group of cells, then we can sample for each cell and for each parameters for the variational distribution, and then we can sample from the normalized counts. We can sample many times, and we can compare the distribution that outputs for a particular gene we can compare it with the other set of cells, the other cell type. And basically what the algorithm do is that it's going to look at the overlap, how much the distributions overlap. And based on this, it's going to give you a score. So it's called the bias factor or likelihood ratio. And it's going to basically tell you, with a lot of evidence, this gene is very different than this one.
Let's go back to the model itself. So now we have the two components. We know, or we can pretend we know what autoencoders are, what variational inferences force is basically approximate way to perform Bayesian inference. Now, talk about the model itself, like the graphical model. What are the essential features of that model? What is the structure of the model?
So for this model, we essentially have a couple of variables. We have some variables that are observed and some variables that are latent. So, for example, we have the observed counts that follow some distribution, and I will explain the assumption on the distribution. We have and we're going to model these observed counts. We have the batch identifier that we'll treat as observed, but we're going to condition on it. And this is because we condition on that some of the other variables are going to be independent from it. We have a hidden variable for the library size. So the library size, you might tell me that this is an observed random variable because it's sum of counts. But somehow we wanted to treat the library size as random and putting a prior on it to be able to regenerate the data. But this is not exactly obvious. We could also treat it as observed. It's kind of a note. And then most importantly, we have this variable for the latent space on which we also need to put a prior because we model as unobserved. And the prior we use for scVI is a isotropic normal prior, which means it's going to try to map all the cells into this cloud of point of which needs to have a Gaussian density. And this we are actually doing some work right now on how to change this prior, and we already did that within other type of models. But yeah, these are basically all the variables that are in the model. And the way, so to finish the definition of this model, we need to define the conditional probabilities or the conditional distributions, which are basically explaining how these variables interact together. And this is very important. And so basically, we define the conditional distribution of the observed counts conditioned on everything else as a zero inflated negative binomial. And so first there is a topic of zero inflation in single cell transcriptomics. And we actually released a blog post about that. So, if you're interested, I highly encourage you to read more about it. But what is the most interesting is that, in the mean of the negative binomial, we use, a neural network that maps the latent space, Z. I mean, yeah, let's call it Z because I've been talking about it for a long time. And maps this value to a vector of size gene expression, but that is normalized in the sense that this vector is going to sum to 1, and then we're going to multiply it by this random variable that we call library size. And so before multiplication, that's what we call the normalized value. And that's, so this is supposed to be independent from library size, and we can use it for differential expression. This is why we don't normalize the data into scVI. And downstream, after the multiplication, we get the observed counts, and the mean of the negative binomial. And this is what you would use if you want to impute data, but by taking into account the library size. And then, yeah, so now we have defined fully what the scVI model looks like.
Right. So if I understand correctly, the trick here is to use a neural network to map your latent space into your almost observed space. So we mentioned before that mapping the latent space to the gene counts wouldn't be a very good idea. So instead you map it to this intermediary space onto which you then apply all these corrections for batch effects, for the library size and so on. And then you also apply like several distributions, like a Poisson distribution or negative binomial or zero inflated negative binomial to get the final counts, right?
Yeah. Yes, that's exact.
And this gives you a function that evaluates the probability or the likelihood of your data, basically, right? That you can then use in optimization.
Exactly. So when I said that we lower bound the likelihood with this variational inference in the resulting optimization bound, what we have is we have basically the likelihood of the prior, the likelihood of the conditional distribution of the counts given the random variable, and the random variable usually we shouldn't be able to know them. But that's why we have this other neural network that are kind of able to guess them. And these are what we call the variational networks. So this other family so there are the networks from the generative model that you just described that from the latent space are able to guess what is the dropout rate. We didn't talk about that, but yeah, there is another neural network to do that. A neural network to guess what are the almost gene expression level or the normalized gene expression level. But there are also the other family of neural networks that from the observed data are able to guess what are the values of the latent variables. And so, for example, what is the value of the latent space and also what is the value of the library size. So those are the main things.
How many neural networks do you have?
So let me see. I always forget. So basically there is because, for each random variable, you need to encode the distribution. So for example, the posterior of the library size given the data is log normal. So you have one for the mean, one for the variance. Then for Z, same thing, one for the mean, one for the variance. Then you have one neural network, there's 4 in the variational distribution. And then you need some neural network in the generative model, as I was explaining. One for the dropout rate and one for the imputed count. So total is like 6 neural networks.
And do you optimize all of these in the same path? So you have all the hidden parameters in the neural networks, and you have are these the same parameters that define your sort of variationally posterior distribution, or do you have a separate set of parameters that define the posterior distribution?
Yeah, so exactly. So on the six neural nets, 4 of them define the variational distribution. So when you write this bound, so there are different ways you can think about it. What some people would do would be to in kind of like a EM algorithm, or variational EM. So you would maximize the parameter of the generative model and then maximize the variational bound and take some steps in one, some steps in the other. But with these VAs, you can actually just take steps over everything at the same time, and it.
But I understand that you took some steps to simplify your objective function, right? So you marginalize away some of the parameters you're not interested in, right? And you do that analytically. Yeah.
So this is for several reasons. So the way I like to describe scVI is by expanding. So instead of talking about a zero-inflated negative binomial, I prefer to decompose this into for the zero inflation, you get a Bernoulli random variable. For the negative binomial, you get a gamma and a compound between a gamma and a Poisson. And to explain exactly which contributes to what, I like to decompose all these zero inflated negative binomial into the parts. And then when you write it, that and I think it's important actually to understand where does the library size gets in, where does the batch effect gets in, etc. And when you write it like this, before deriving, you can, because you cannot make inference over these discrete random variables. It's really difficult to have some discrete unobserved latent variables with these VAs. And so, for example, the zero inflation, whether it's active or not, it's not exactly interesting for our purpose. What is more interesting would be what is the probability of zero inflation, or at least that's already enough of an indicator. And so that's why we integrate these variables out, and we get exactly what I was describing before, which is this zero inflated negative binomial distribution. And sometimes it's easier to present it the latter way. And when you perform this type of integrals before inference, so this analytic integration, it's exactly called collapsed variational inference.
And since you mentioned zero inflation a few times, let's talk about that a little bit. So I think it's a somewhat controversial topic because a lot of people tend to agree that it exists, but then some disagree. So I remember there was a blog post from Valentine Svensson where he said that there is sort of no evidence for zero inflation. I think that was specific to a specific single cell sequencing protocol or system. Then there was a very recent preprint, I don't remember by whom, where they said that basically, there's no reason to zero inflate. And I think you recently, or your lab recently published also a blog post on this subject. So what do you think? Is zero inflation necessary? Is it real, or do people sort of overestimate it and its effect?
So thank you for asking. So first, I would like to give a disclaimer that I am not working with the experiments myself, and so I am not exactly an expert on how you would give experimental, intuition about what is zero inflation. And I think it's fair for people to be quite skeptical about that. However, I can talk about from the computational perspective and from a statistician's side that given data, we can basically make a hypothesis testing, and we can give some evidence. And so what we did for example, the previous blog post that Oscar and Pierre worked on here in the lab, we try to understand, okay, let's take some data set. For example, let's fit scVI with a negative binomial distribution, and then scVI with a zero inflated negative binomial distribution. And let's see which one wins. So and there are a lot of different ways to do this in a principled fashion. It's called model selection. And what we see is that so Valentine Svensson tried to work with these negative control data sets with ERCCs, which are like basically, I'm not sure it's even synthetic RNA, but it's like, basically these are not real cells, and in a sense. And we try to see what can happen. And basically on these synthetic data sets, or on this ERCC negative control data sets, we find that they are actually negative binomial, which means that there is evidence to say that negative binomial is a better model to describe this than zero inflated negative binomial. However, something a bit striking is that when we took on our code base, we took like 5 or 6 data sets we had, and we see that it's the opposite. So whenever you have some real cell types and some real biology, it looks like zero inflation provides actually a better way to explain the data. And what this means is that it completes a little bit what Valentine was trying to explain in his manuscript, which says that there is no evidence that on the technical side, in what happened in the experiment, there is no technical effect that gives rise to zero inflation. However, there might be some evidence that, biologically speaking, there is a process that is happening which is similar to, which is giving rise to zero inflation in the data. And we're actually investigating this a little bit more. We don't have clear hypothesis, but we would like to investigate whether this zero inflation would be just explained by just basically some model of transcriptional bursting, in which you have some intrinsic noise in gene expression, which inside a cell, the number of counts actually describe a hypergeometric distribution or a beta Poisson model. And in that case, depending on the kinetic of the promoter, whether the gene is on or off, you can actually have some like two modes for the gene expression. And maybe because of limited sensitivity, the lowest mode gets just squashed down to zero, and that's what is this zero overabundance might be. So until here is very conceptual, but this is what we are trying to understand.
Very cool, and we'll link to the blog post, to that blog post as well. And another interesting aspect of your model that you mentioned is its scalability, meaning that it can process the larger amounts of data more efficiently than the alternative methods. And that's a bit counterintuitive because your model is not a very simple one, right? I think some of the earlier models were much simpler, didn't have as many neural networks, and yet you not only made the model more expressive, more complex, but also improved the scalability. How is that possible?
So first, there are different bottlenecks when you design an algorithm, and from the pure computational perspective, you want to ask the question, how am I going to implement my model? So depending on which language you use, or depending if you interface with, with C or C++, you might have some huge speedups. So that's the first thing. It's a little bit less, not naive, but a little bit more on the statistical side. Most of the inference nowadays with machine learning can be written as an optimization procedure. So yeah, so if you use sampling and you have a lot of data, most of the time, or if you don't, depending on the sampling you use, it might take a bit more time. But let's consider only optimization. When you write an optimization problem, you want to guess how many variables am I using? And basically, if you have a number of variables that scales linearly with a number of samples, it means that it's going to be very expensive in terms of memory to just to fit your model. So you're going to have some memory limitations. And even if you have tons of memory, it still might take a lot of time. If, for example, you do full batch. So here, batch is not biological batch. It just means the way you optimize your data set. Full batch means taking a gradient path through all the data, which means you would average all the data points and take a gradient. So if you take a gradient step, or if you take a second order optimization step on all the data set, it's very expensive computationally. And so you can gain some significant speed up by using some stochastic optimization. And the neural networks literature and this VA literature is a way where you can, in the principled way, use a stochastic optimization. It's called stochastic variational inference. And because of all this sampling from the variational distribution and subsampling from the data points, we can actually do something that is considerably faster. And so there is this, and then there is also some other method. For example, we'd consider in terms of memory footprint a full cell by cell similarity matrix. And for this type of calculations, you have n square complexity memory. And so it's going to be a nightmare to scale this to 1 million cells. So these are, I think, I try to explain the main bottlenecks with computations. And so basically, even if scVI looks like a more complicated model, because of these tricks that we used, stochastic optimization and like amortizing inference, which means not learning a parameter for each data point, but parameterizing with a neural net, which is very standard in VAEs.
Pretty cool. And in terms of the technologies, what do you use? You mentioned the language plays a role. So what language do you use? What libraries for optimization do you use?
Yeah, so here there is nothing very original with respect to the culture of these VAEs and neural network literature. So our code base is based on Python, and we rely on deep learning libraries such as PyTorch. So all the code base is in PyTorch, and we found that it's a very flexible tool for research. And I was actually surprised to see that, happily surprised to see that there are some other like single cell people that start using these tools even for fitting linear models. And they use stochastic optimization and then can do a lot of interesting things even with simple models, but start using the tool of at least stochastic optimization that can allow for more scalable inference. For example, a model in which I would like to give a little bit of credit from Sandrine's Dudois lab in Berkeley, which is called the ZIMBI Wave, which is actually probably the first model to have used this. In single cell data, to my knowledge, this zero inflated negative binomial distributions, and the way they were fitting the model. So first, I think the model was written in R, and it was using some second order optimization method on the full data set. And so these steps are really expensive, and this made the model to be very slow when you had a lot of genes and a lot of cells. And so in this way, that you could have recoded all ZIMBI wave with the stochastic optimization and stochastic variational inference, and it would have been probably as fast as a scVI. I mean, even maybe faster as.
And for variational inference, does PyTorch have anything sort of built in for that, or did you have to implement it on top of PyTorch?
So there are some kind of a subroutine for distributions. So you can, but it's not. It's actually kind of short to write. I think when I wrote the first, to give you an insight, like the first code for the scVI manuscript, the one I used for the paper, I think the main subroutine for fitting the model was like 80 lines of code in TensorFlow.
Wow, that's very impressive.
80, yeah. So it's really not a lot of code. And the main part of the code was like, defining this zero inflated negative binomial likelihood. And then all the rest is kind of pieces of code that are already used in VAEs and even AT is just really small, yeah. So then the rest was like all data loading and stuff. And the reason for which the database now is like pretty big is that we want people to just come in, plug in, and if they want to change zero inflated negative binomial by something else, then they just can, and that's what we would like to provide for the community also.
Sweet. And so the last technical aspect of your work that I want to get to is the hyperparameter optimization that we talked before. And we talked before about it from the perspective of the user, but now I want to consider from the perspective of the implementer. So Gabriel, I believe you did that work. So how does this thing work?
Oh yeah, thanks for asking. So just a disclaimer, I interfaced scVI with a library that actually takes care of implementing the technical aspect behind this hyperparameter optimization process. But still, I can give some insight, some technical insight on how exactly hyperparameter research works and how we chose to do it. So first of all, the context is as follows. So you have your model and a specific metric and your set of hyperparameter or a space of hyperparameters you want to search in, and you want to optimize your metric. So in order to actually get one observation point, you're going to have to go through a full training of your model, and that can be very expensive. For instance, with scVI, training can range from a few minutes to over an hour if you have a very large data set, as Romain mentioned.
And for scVI specifically, what hyperparameters are we talking about?
The search space, if I remember correctly, is model architecture. So number of hidden layers, number of hidden neurons in those hidden layers, dropout probability. There is also the learning rate of the optimizer.
And so when you say the dropout probability, there are
I think there are two different dropouts. Yeah, not to be confused with the dropout in the graphical model, scVI's graphical model. This is the dropout probability for the dropout layers within the neural network. With dropout is a standard process that is used to perform regularization in neural nets. So yeah, not to be confused with scVI's dropout event. And that's it for the search space, I think.
Mm-hmm. And are these parameters per neural network? Does each neural network have its own parameters?
So for now, the way scVI is implemented, there's a common ground for these parameters for every neural network. So they will affect all the neural networks within scVI. But we're working towards making each of most of the neural networks fully independent of each other parameter-wise. But yeah, as you said, for now there are common parameters for all of the neural networks.
Got it. Okay, so how does the magic happen?
So again, disclaimer: I'm not the maths are absolutely not mine, and the methods that we use have been existing for a long time, and we rely on a library that implements it in itself. But so quickly, to perform a hyperparameter search, the simplest methods are a grid search or a random search. For the former, you're just going to fit a regular grid on your search space, and then sample, and then go over that grid and test each tuple of parameters and observe the metric. And then in the end, you're going to select the best set. And for random search, you just randomly sample. You give yourself a budget, a number of evaluations, and you randomly sample the search space. So these methods are very, very costly, and they scale exponentially with the size of your search space. One option you have to kind of be smarter about this is to use Bayesian optimization, and that's what we do in the scVI tool. So basically, what happens is you state a surrogate model for this expensive process that is training your model and observing the metrics. So, for example, this is going to be a Gaussian process that is going to represent your belief on this black box function, as we could call it. to be clear, this function I mentioned, you could call it f, is the f that gives you the metric, the observed metric, given a set of hyperparameters. So you give a surrogate model for this function f, which represents your belief about it. So that is often a Gaussian process. And then you use what we call an acquisition function that relies on this model of your black box f to suggest new trials given a specific criterion. So, for example, you're going to try, given this Gaussian process that mimics your black box f, you're going to try to maximize the expected improvement of the metric for the next trial that you're going to do, that you're going to process. And that's the way you're going to generate new trials. And usually these acquisition functions are responsible for setting the trade-off between how much exploitation are you going to do versus how much exploration you're going to do. And by exploitation, we mean that continuing to sample the regions where you know that you're going to obtain a good metric compared to what you've already observed. So, for example, if you've been searching over your search space and you've done some trials with a high learning rate and others with a lower learning rate, and you notice that with a lower learning rate you get a better metric. Exploitations means continuing to sample towards this low learning rate region. Exploration, on the other hand, is sampling the regions where you have a low confidence on your density estimate, on what you think, on your belief. So, for example, if you in between those two learning rates, you have an intermediate value, and you have not used that value often, and you don't have a lot of observations that have been generated using this value, you have a low confidence on the value you estimate for this specific learning rate region. So exploration is going to entice you to generate new trials in this area.
Right. So low confidence essentially means very high variance, and so very high potential for huge upside.
Yeah, exactly.
All right. It's very interesting. Before we wrap this up, do you want to talk about any new developments coming from your lab? So scVI, how old is it exactly? It's been like 3 years?
So I started working on scVI summer 2017.
Okay, so 2 years.
2 years, yeah, two big years.
Doesn't sound too long, but it feels very long in such a dynamic space, I guess.
Yeah. I guess single cell is such a fast field that indeed it seems already long. So this has been if I can spend a little bit of time giving credit to the whole team. We've been very lucky to have been working with a lot of persons overall on this big project that is scVI. So first, I started scVI being mentored by Jeff Regier, Nir Yosef, and Michael Jordan, who are all now. Jeff is also a faculty, so we're all faculties. I'm being advised throughout my PhD by Nir Yosef and Michael Jordan. We've had some graduate students that joined the team, Adam and Chenling, that has been working on total VI and scVI. I will explain later. And we've been very lucky to have amazing master students visiting from France, from either École Polytechnique or École Normale Supérieure. So this year we have Gabriel, Achille, Pierre, and Oscar. Last year we had Edouard and Maxime. And we also had some really amazing undergraduate students that came last year, so Jules and Yening. And this has been very fun to have this overall very social project in which we try to work all together. So about the new model we try to work on, for example, there is scVI, in which, so I was talking earlier about the prior on this latent space. And basically, scVI is an extension of scVI in which this prior is extended to be a Gaussian mixture model. And this is very interesting because in this scenario, if you know for some of the cells, you know some of the cell types, then you can basically it's very simple with this graphical model to do some semi-supervised learning, which means you can extend this to the scenario of I have one data set that for which I know the cell types, one data set for which I don't know, and then you can get a full posterior for all the cells that you didn't observe the cell types. You can have a distribution of like which cell belongs to which cell type with which confidence. And you can also use it in a scenario where, for example, you have a tissue for which the cell types are very, very hard to distinguish, for example, T cells, and you have some markers for subcell types of T cells, but you don't know exactly which are which, and you can use these markers to have to label a small number of cells, maybe 10, 20, but very confidently, and then you can leave it to scVI to classify the rest of the cells. So this is available on bioRxiv and now on our code base. Then we also worked, so scVI is work with Chenlin. Then there is a GimVI. So GimVI is for gene imputation. And so basically here we try to work with trying to harmonize and to make work together spatial transcriptomics with single cell transcriptomics. So for example, for fish data, you can observe just a certain panel of genes that is defined beforehand. It makes sense that if you have single cell transcriptomic data for the same tissue, and but not the same cells, right? Just the same tissue. And then what you would like to hope is that if you try to impute a gene that, in the sense of the covariance matrix, that looks a lot like some of the genes in the spatial panel, then you're going to be able to impute it for all the cells, and you can visualize it on the spatial tissue, and you can detect whether this gene is spatially differentially expressed or not. And this for this line of work, I refer to the work of Valentine Svensson, for example. So this has been work with Achille and with Maxime and Jules. And then there is a total VI that is still work in progress with Adam. And in total VI, we're trying to build a model for total-seq, or what was also called side-seq. Yeah, exactly. So and we try to build the joint model for the protein measurements and the gene expression. So basically, side-seq is an extended single-cell RNA sequencing protocol in which you can add antibodies with poly-A tail that is going to bind to different proteins, protein markers on the surface, and you can observe. And doing this kind of model is interesting because you're able to in an unsupervised way to learn in which cell which protein have bounded or not, because you have a lot of background, and the distributions are actually pretty interesting. And so we're able to disentangle the background for the foreground and understand which proteins are binding to which cells. And so this is still work in progress. But whenever we have an algorithm we are happy with, we will also put it on the code base.
Pretty cool stuff, guys. And it's very inspiring that in such a short time you've managed to build this tool, which is now pretty much the classics and the go-to method for so many researchers. So thank you for coming on the podcast and sharing your perspective on this.
Thank you very much for inviting us. Research sometimes can be a bit dry, and reading a paper is not might not be the easiest thing to do, and I think it's a very inspiring exercise for me and Gabriel to be here today and try to dig a little bit into the work and give it some details, but also high level. So we're really happy to do that. And thank you very much, Romain, for inviting us on the podcast.
Yeah, I completely agree with Romain. Additionally, I'd like to mention that it's also a lot of fun to be on this podcast with you, Romain, and that you were very helpful with setting this up. And I definitely recommend other researchers to come here and talk about their research and how cool it is. Thank you.